Ladies and gentlemen, thank you so much for joining GenScript webinar today. I'm your host, Bunhua. Today, we are going to discuss how to overcome timeline challenges in antibody drug discovery. GenScript is a global leading technology and service provider of life science R&D and manufacturing. Built upon its solid gene synthesis technology, GenScript is divided into four major platforms, including the life science service and product platforms, the biologics contract development and manufacturing organization platforms, the global cell therapy platform, and the industrial synthesis biological product platform. Our speaker today, Dr. Hui Fun Tan, is the regional product manager in GenScript. She has 8 years of research experience in cell and molecular biology. Her previous research mainly focuses on understanding how a family of proteins known as kindlings regulates the stability of mitotic spindle assembly in addition to their primary role as an integrins regulators. Understanding the mechanism behind its functions in cancer to their primary, it helps to identify the molecular targets for therapeutic purposes. Before we get started, here are some tips for you. On your screen, you are able to see a Q&A box. Feel free to ask any questions about the presentations. Our speaker will answer your questions after the presentations. There are also downloadable resources that help you better understanding our offerings. On the bottom right, there's a survey questions to help us better understand your needs. Please help us to fill that while watching the webinar. So without further ado, I'll pass the stage to Dr. Tan.
Yeah. Okay. Thanks, Bunhua, for the introductions. Hi, good morning. Good day, everyone. I'm Hui Fun, the product manager from GenScript. Thanks for taking time to attend today's webinar. It is my pleasure to be here to share how to overcome the timeline challenge in antibody drug discovery. In today's webinar, I will first talk about the overviews of antibody drug discovery and its development process. After that, I will share some of the lead optimization strategy in antibody drug development. Lastly, I will discuss how to overcome the timeline challenge by using our GenScript antibody discovery and expression platforms. First, I will give an overview of antibody drug discovery and development. It has been more than 3 decades since the first monoclonal antibody immunomodulant CD3 was approved by US FDA in year 1986. The increasing importance of therapeutic antibodies is apparent, as therapeutic antibodies has become the predominant treatment modality for various diseases over the past years. Due to the recombinant antibody technology's advance has made the discovery and development of monoclonal therapies quicker and more efficient. After that, more and more therapeutic antibody has been approved and come out into the markets. And the global therapeutics monoclonal antibody market is expected to generate revenues of 300 billion by 2025. In last year, May, 100 therapeutic antibodies has been approved by FDA, and majority of them are in cancer therapy. In recent years, traditional immunoglobulin antibody has been further engineered for better efficacy and safety, and the technological developments in the fields enable the design and productions of engineered antibodies capable of mediating therapeutic functions hitherto unattainable by conventional antibody formats. Recombinant antibody can come in different formats, including canonical and IgG-like antibodies and bispecific antibodies. Antibody basic structures is a Y-shaped molecules consists of heavy and light chain folded into constant and variable domains. The FAB domain consists of the 2 variable and 2 constant domain. The variable domain FB contains the 3 hypervariable loops known as the complementarity determining reagents, CDRs, and framework regions. CDRs that allow an antibody to recognize an unlimited numbers of antigens. The constant regions at the track of the antibody include the site involved in the interaction with other components of the immune system. With the current biotherapeutic market's dominance by the antibody molecules, bispecific antibody represent a key component of the next generations of antibody therapies. Bispecific antibody format include a full antibodies size containing a Fc regions, antibody fragmented size without the Fc regions such as the scFv, FAB, and so on, and the complex larger than the antibody format including the bivalent and multivalent formats. Bispecific antibody can target two different antigens at the same time, such as simultaneously binding tumor cell receptor and recruiting the cytotoxic immune cell. Structural diversity has been fast growing in the bispecific antibody field, creating a plethora of the novel bispecific antibody scaffold, which provide a great functional variety. Through several decades of developments and explorations, therapeutics antibodies has become the most widely used and approved therapeutic methods in clinical practice to treat various diseases. A drug discovery program initiated because there is a disease or clinical conditions without a suitable medical products available, and due to the unmet clinical needs, which is the underlying driving motivations for the projects. The intention drug discovery process ensures to find a drug like small molecules and biological therapies that will progress into the preclinical trial, if success into the clinical trial developments, and ultimately to be marketed medicine. The overall discovery process of therapeutic antibody can be broadly divided into four stages, which are the targeted discovery, target generations, lead validations and optimizations, preclinical candidate selections. The process of discovery and development of first-in-class drug begins with the identification of a new drug target, such as enzyme linked to a disease process. Once a new target has been identified, various strategies are used to provide validations of the targets and to support the decisions, such as initiating an extensive drug discovery program, conducting a proof of concept trial in humans, or partnering with another organizations. Different phases of the drug discovery process involve different strategies and design. In target discovery, therapeutic targets are identified and characterized through different approaches like bioinformatics, genetic associations, expression profile, functional screening, and so on to demonstrate how its modulation affects disease and review its direct involvement in the disease. Through genome-wide association study to identify the gene associated with the particular disease, scientists have to perform in vitro and in vivo assay to identify the targets. Besides this, X-ray crystallography is utilized to determine the 3D structures of the protein ligand complex, as well as some other tools and criteria to validate crystal structures available in the database. The crystal structures of the protein ligands complex analysis allow to study of the specific interactions of a particular drug with its protein target at the atomic level. It helps to design and improve the drug. In the target generation phase, validated antigens targets are used to generate therapeutic target antibodies candidates by eliciting an immune response through antigenic presentations. Scientists have to perform high-throughput screening to define and execute strategies for evaluating non-cross-reactive antibodies. Through structure-based drug design and molecular dynamic analysis to develop binding and functional assay and select the lead molecules with desired functions and favorable biochemical and biophysical properties to make sure the in vitro safety assessment and in vivo efficacy as well. In the lead validations and optimization phase, scientists would like to improve the lead safety, efficacy, and developability, which I will discuss further in the next sections. Through medical chemistries or rational drug design strategies to optimize drug-like properties and also perform the absorptions, distribution, metabolism, restrictions, and toxicity studies to validate and optimize the lead. Once lead candidates are identified. Preclinical tests are required to evaluate their safety. Preclinical trials are conducted on non-human subjects to study the efficacy, toxicity, and pharmacokinetics of the therapeutic antibodies. This is the last antibody discovery stage before moving on to clinical trial studies. In this phase, in vitro and in vivo assays are carried out to ensure the safeness of the candidates, commercial assessment, legal evaluations, clinical biomarker plans, regulatory plans, manufacturing plans, and patients' certification plans will be designed before moving on clinical trial. Next, I would like to discuss what are the challenges which scientists and researchers face nowadays and what are the lead optimization strategies scientists have used in the antibody drug development. Antibody drug discovery is an arduous process that often starts with the extensive screenings of antibody library to identify optimal candidates, namely those having high affinity antigens, bindings, and other desirable functional properties. There are some of the challenges which scientists encounter nowadays on the antibody drug discovery route. Antibodies are expensive to produce because of the difficulty of process of isolating and quality testing thousands and thousands candidates. This process can take up to years and require lots of technical experts to synthesize one therapeutic drugs. Therefore, timeline and financial issues are always the main problems in the drug discovery and development. Other than that, manufacturing issues such as instability of the antibodies, undesirable byproducts from the manufacturing process, yield and quality inconsistencies, or unable to reproduce the products also will lead to discovery process delay. Appropriate targets screening and selection methods are required to ensure the targets and leads are correctly selected before further validations. Insufficient validations of drug targets at an early stage has been linked to costly clinical failure and low drug approval rate. Besides, antibody drug developability, safety, and efficacy play crucial roles in antibody drug discovery. These involve several optimization strategies and methods to improve the final drug candidates. Without a proper drug optimization and modification step, many potential candidates are unable to pass through the safety and efficacy tests. Therefore, select a proper algorithm design is important. The field of therapeutic antibody is becoming increasingly competitive, whereas the development cost of successful therapeutic antibodies is also increasing with time. Therefore, identifying novel methods to optimize the safety, efficacy, and manufacturability of the antibody candidates would be critical to the efficient development of therapeutic antibodies. Therapeutic antibody undergo 4 major sequential steps during the preclinical development, namely targeted research, targeted generations, leads validation and optimization, and preclinical enability, which I mentioned previously. In these sections, I'm going to discuss further on what are the methods scientists use to perform antibody lead optimizations. Developments of the therapeutic antibody usually go through multiple steps of optimization process in order to reach the best balance on safety, efficacy, and manufacturability and have the subsequent clinical success. In addition to traditional antibody engineering technologies, newly emerging computer-aided approach are evolving the ways the therapeutic antibody leads optimization process are performed. The strategies of humanizations, deimmunizations, and tolerization are performed to enhance the safety, and whereas the affinity maturations and Fc effective function improve are performed to enhance the efficacy. The pharmacokinetics improvement and pharmaceutical property developments are performed to improve the developability features of the antibodies. Antibody humanization is usually performed by studying the difference between the non-human antibody sequence and that of its human homologs. The purpose of doing this is to reduce the immunogenicity by generating the chimeric antibody through fusions of humans antibody constant region with variable regions of non-humans antibodies, and modify the variable regions to increase the similarity to antibody variants produced naturally in human. Computer-aided design, phage display, and ease display are the three widely used approaches for humanization. Computer-aided design allows the user to construct a 3D homology model structures, analyze the structures and design mutations in silico, and phage display and ease display allow them to physically assess all possibility to determine whether a mouse or human amino acid residue should be selected at each testing position. While numerous factors such as aggregations, dose, route, and targets can contribute to the immunogenicity of the therapeutic antibodies, one of the key contributors to this effect is the epitope sequence content within the antibodies. Deimmunization is the process of the identifying and removing these epitopes through combinations of information from linear sequence predictions and percentage of the displaying of epitopes from 3D models is a better way to identify immunogenicity epitopes. Other than that, removing immunogenicity epitopes of a therapeutic antibodies candidate, another way to repress its human immunogenicity by inducing the T-cell regulatory epitopes in the antibody functions will actually cause immune tolerization. The next step in the therapeutic antibody optimization process is to improve the antibody efficacy. Affinity maturation is a route method used for this purpose to improve the binding of an antibody to its target antigens. Method used for antibody affinity maturation including random mutagenesis, targeted mutagenesis, chain shuffling, and in silico approach are widely applied. The first three methods are usually performed by using a display technology, such as a phage display. In silico approach like bioluminescence is based on computer-aided design. Besides, Fc gamma receptors on their surface are capable in recognizing and binding to the Fc domain, and as a result regulate antibody-dependent effector functions by the cells. Employing the computational design algorithm, glycosyl modifications, and high-throughput screening to improve the Fc effector functions. Improving the pharmacokinetics of a therapeutic antibody allows its dosage to be lower, enable a subcutaneous formulation to be developed and the cost to be reduced, improve the clearance of a therapeutic antibody from the body through Fc engineering, and lower the PI of the antibody can reduce the non-specific clearance. For the antigen binding mediated clearance, possible selected the pH-sensitive antibodies from the library of the antibodies variants and humanization variants, or else rational introducing of the ionization group in the protein-proteins interface to reduce the antigens binding mediated internalizations and clearance. Desirable pharmaceutical properties of a therapeutic antibody including high thermal stability, high solubility, high chemical stability, and low heterogeneity. These properties contribute to the successful retention of biological activities of the antibody during storage, minimizing aggregation for less immunogenicity, improving production efficacy, yield to reduce cost of goods, enabling high concentration formulations, and facilitating good quality control in manufacturing. Poor thermostability may cause antibody aggregations and low expression. To improve the thermostability of an antibody, hydrophobic core and charge cluster residue need to be optimized. Library-based site mutagenesis study, CDRs crafting onto non-antibody frameworks, consensus design in antibody variable domain, and computer antibody structures modifications are used to optimize antibody thermal stability. High concentration of the formulations is important for applications of the therapeutic antibodies in patients, especially for the treatments of chronic disease. Removal of the surface hydrophobicity is a major approach for improving antibody solubility. Structures-based engineering, including altering the PI and reintroduce of the N-linked carbohydrate moiety into the CDRs, and combination of the structure-based design and somatic variance optimization can also be used to improve the solubility of the therapeutic antibodies. A lot of chemical degradation reactions can affect antibody stability and lead to reductions in the potencies of the therapeutic antibodies. To improve the chemical stability, targeted site mutagenesis via a phage, yeast, mammalian library, or computer-aided design is performed. Therefore, a best-in-class strategy may require the optimization of all these properties to generate a good therapeutic antibodies. Here, I would like to share a case study in antibody lead optimization, which modifying the Fc effectors of the amivantamab to increase the binding affinity of Fc gamma receptor and enhancing the ADCC activities. Amivantamab is a fully human bispecific IgG1 antibody targeting both EGFR and cMET. It induced a natural killer cell-mediated and antibody-dependent cellular cytotoxicity and tumor cell internalization with macrophage and monocyte, resulting in the phagocytosis of the EGFR and cMET receptor from the tumor cell surface to the immune cell surface, and thus down modulations of the signal transductions and downstream pathway. Evidence from multiple in vivo models suggests that antibody binding to the cellular Fc gamma receptor is the major pathway for antibody-dependent cytotoxicity. In this study, scientists have identified low fucosylated bispecific antibodies targeted cMET and EGFR enhance the binding affinity to the Fc gamma receptor 3A. The effect of low fucosylated on the antibody binding to the Fc gamma receptors was evaluated using an alpha screen competitive binding assay. As shown in the figures, and low fucosylated bispecific antibodies had enhanced the binding to the Fc gamma receptor 3A compared to without fucosylated modifications bispecific antibodies. Besides that, low fucosylated bispecific antibody was most potent and reached the highest efficacy in non-small cell lung cancer and the gastric adenoma carcinoma cell line. In the experiments of the in vivo female nude mice studies, nude mice bearing a subcutaneous non-small cell lung cancer tumors were treated with the same antibody panels. The strongest tumor growth inhibitions was observed with the low fucosylated bispecific antibodies based on the efficacy study using the tumor model, driven by both EGFR and cMET review that the mechanism of actions of low fucosylated bispecific antibody include both inhibition of the cMET and EGFR pathway. Lastly, 24 hours following the second dose of the low fucosylated bispecific antibodies on the non-small cell lung cancer tumor was actually harvested, and pharmacodynamic effects were assessed by the Western blotting, and level of the total and phosphor EGFR and total and phosphorylated cMET were significantly reduced in the tumor from the mice treated with the low fucosylated bispecific antibody compared to those treated with the vehicle control. In conclusion, through antibodies optimization strategy to modify Fc effectors of targeted antibody to low fucosylated form, which actually enhance the ADCC activity via its increased binding to the Fc gamma receptor 3A. Lastly, I would like to share how to overcome the timeline challenge which scientists or researchers encounter in antibody drug discovery finding by using our GenScript antibody discovery and expression platforms. The rapid pace of the scientific advance is enabling a greater understanding of disease at the molecular level. In turn, scientific, technical, and regulatory challenges related to the drug development create the complexity as companies often focus their R&D where the science is difficult and the failure risks are higher. As a result, the process for researchers and researching and developing new medicines is growing in difficulty and length. On average, it takes at least 10 years for medicines to complete the journey from the initial discovery to the marketplace, with the clinical trial alone taking 6 to 7 years on average. The average cost to research and develop each successful drug is estimated to be 2.6 billion. This number incorporates the cost of failures, which include the thousands and sometimes billions of millions of the compounds that may be screened and assessed early in the R&D process, only a few of which will ultimately receive approval. The overall probabilities of the clinical success is estimated to be less than 12%. Success requires immense resources, which include the best scientific mind, highly sophisticated technology, ever-evolving manufacturing process, and complex project management. In the last section of this webinar, I shall focus on how does our GenScript help scientists and researchers in overcoming the timeline challenge through our proprietary antibody generations platform. Just a brief overview of our GenScript service. We actually provide a series of life science services and products such as gene synthesis, oligonucleotide synthesis, peptide synthesis, protein productions, antibody development, and research reagent and equipment catalog product as well. These are highly utilized in diverse applications, for example, basic research, proteins and antibody engineering, biopharmaceutical, gene and cell therapy, synthetic biology, and diagnostic field. We have been improving our clients' competitiveness through providing our superior quality, fast delivery, and cost-effective service and products. We would like to contribute more of our efforts to speed up the evolutions of the whole biotech and biopharma industry to realize multi-win among all the participants partners in the industry. Therapeutic antibody has become the predominant class of the new drug developments in recent years. With GenScript solutions, we aim to support your antibody drug discovery and development via a simple but specialized process. We have integrated a series of precise and consistent scalable solutions to facilitate each principal milestone in antibody drug discovery, combining speed and quality towards investigations new drug applications. For antigens generation, antibody discovery, and antibody lead optimization part, GenScript Life Good is able to help you from the gene synthesis, mutagenesis, peptide service of those upstream service to downstream service such as AC cell line generation, antibody discovery, and antibody and protein expressions. Beyond that, our sister company called GenScript ProBio is able to provide a CRO and CDMO service as well. In today's talk, I will mainly focus on two of our life science group service, which are the antibody expression and generation in the rabbit monoclonal antibody service and our proprietary TurboCHO antibody expression platform. First of all, GenScript is the award-winning custom antibodies provider with over 15 years of experience and successfully delivered 50,000 antibodies. We have proprietary technology to not only cover antigens design, but also discovery platform and beyond. In the antibody discovery process, we provide one-stop solution we start with the antigen design and productions, and then immunize the animals with the antigens to generate the antibodies. Afterwards, we screen the antibody with different characterization assay. We can also provide antibody production downstream and other service as well. Beyond than traditional murine hybridoma monoclonal antibody generation, we do provide rabbit B-cell cloning and beacon single B-cell platform. In rabbit monoclonal antibody generation, we isolate the peripheral blood mononuclear cell directly from the rabbits, and then under sterile condition. Then afterwards, we go through the memory B-cell enrichments, B-cell stimulations, cell culture and screening, and then ultimately sequencing and antibody productions. I shall discuss further with our beacon single B-cell platform in the next few slides. What's so special about monoclonal derived from rabbit hosts? Rabbits has a robust and unique immune system. Rabbit-generated antibody provide high specificity, affinity, and more diversified antibody compared to the rodents. Let's have a closer look on the difference between rabbits and mouse-generated monoclonal antibodies. In B-cell development and antibody maturation process, rabbits have additional VDJ recombinants, gene conversions in appendix and GALT compared to the mouse. In antibody affinity is relatively higher compared to the one generated in the mouse. Rabbits had also only have one subtype IgG, which compared to the mouse have IgG1, IgG2A, IgG2B, IgG3, and IgG4. Rabbit IgG have more disulfide bond, which can have a good stability and good for storage and heat. Other than that, rabbit-generated antibody can recognize diversified epitope compared to mouse. The antigen-presenting cell in rabbits have many CD1 isotypes, which create a robust immune system of the rabbits. Other than that, rabbits is able to respond strong immune response against small molecules and heptans, and it is uncommon in rodents. Rabbit-generated antibodies are able to cross-react with mouse autologs of humans' antigens. However, mouse is limited and not good to recognize rodent proteins. Ultimately, rabbit-generated antibodies can use in many applications, such as Western blot, ELISA, immunoprecipitations, immunofluorescence, immunohistochemistry, immunocytochemistry, but mouse-generated antibody are not well used in the immunohistochemistry and immunocytochemistry. In typical hybridoma workflow, normally require 12 weeks to perform the culture preliminary screening, single cell cloning and expansion of clones, and then perform the validations assay to check the antibody expressions. However, in MonoRab beacons platform, it is just need a day to shorten the whole process from 12 weeks to 1 day. Beacons optofluidic platform provide the highest probabilities of achieving your ideal monoclonal clones in shortest amount of time. Single B-cell cloning or sorting technologies is a popular microsystem based on screening methods that is increasing being adopted throughout the field of antibody discovery. The Beacon platform by Berkeley Lights harness the latest optofluidic technology and combine the isolation, screening, and evaluation of B cells on the chips with the integrations of high-throughput platform for assay screening. Supernatant harvest after the B-cell enrichment and stimulation, then load on the chips of Beacon's machine. The chip consists of thousands nanopens channels. This software automatically identifies single cell and direct them into nanopen channels all at once. So chips are loaded with the cell in less than 30 minutes. Each well have a specific ID numbers, and it can run sequential and multiplex assay with fluorescent laser detection to validate the B-cell specificity and affinity in a very fast pace. After that, the data and specific positive B-cell can be exported and proceed with the downstream sequencing and antibody productions. Here, I would like to share one of our in-house express case study to discover rabbit's monoclonal antibody, which recognize SARS-CoV-2 spike receptor binding proteins by using our MonoRab beacon single B-cell screening platform. After rabbits immunized with the SARS-CoV-2 spike proteins, PBNC was collected and we perform a memory B-cell enrichment. After that, B-cells are loaded on the chips of beacon machine to perform an antigens positive screening, which I have discussed earlier, and this can be done within a day. In this case study, we are able to get around 142 antigens positive hit. In summary, on the S protein IgG affinity testing, rabbit monoclonal antibody has 10 to 100 times higher affinity than mouse-generated monoclonal antibodies. Other than that, beacon-generated monoclonal antibody has highest diversified, which fall into 4 different epitope bin compared to the one generated from the monoclonal antibody hybridoma generated monoclonal antibody, which are highlighted in purple. They are actually fall within the same epitopes. Besides, these antibodies for SARS-CoV RBD protein generated from Beacon's platform are functional. They are validated in the pseudo-neutralizing assay, and they are able to detect the targeted RBD proteins in the Western blot. Antibody towards the RBD hits were also validated using a FACS. Rabbit monoclonal antibody can detect the RBD overexpressing cell line with good specificity. This antibody generated from the MonoRab beacons platform with different epitopes bins form a good pairing for ELISA kit development. The sensitivity is 20 picogram per mil, which is pretty high. It can also detect the pseudovirus. In conclusion, through MonoRab beacon single B-cell service, which can help to shorten your antibody discovery and generation from originally 5 to 7 months for traditional hybridoma technology to current 2 to 2.5 months for this MonoRab beacons express service. This platform is not only helping you to save time, and it also increases the success rate of your positive antibody hit generation. The success of the antibody therapeutic is strongly influenced by their multifunctional nature that couple the antigens recognized mediated by their variable regions with effective function and half-life extension mediated by a subtype of their constant regions. Monospecific IgG formats is not optimal for many therapeutic applications, and this has led to design of a vast of unique multi-specific antibody formats that enable targetings of multiple antigens or multiple epitopes on the same antigens. The first monoclonal antibodies murine CD3 was approved by US FDA in year 1986, and afterwards more research was done to overcome the limitations of the monoclonal antibodies. Many of others antibody formats have received market approval, and more than 110 types of bispecific antibodies are at various stages of clinical trial. In drug screening and discovery, sometimes synthesize an adequate amount of antibodies targets within a limited timeline is not an easy task for researcher. In the next part of this webinar, I would like to share how does GenScript proprietary antibody production platform help our scientists to overcome the timeline and generation issue. In this year, February, we have newly set out our proteins, antibodies, and gene synthesis production site outside of China and US. It is located at Asia Pacific region, Singapore. The state-of-art of Singapore protein production facility are we can produce in the high yield and high purity of proteins and antibodies from milligram to kilograms. It has faster turnaround time, which just need as short as 2 weeks, including gene synthesis to typical antibodies. We have a full range of QC and characterization tests available to further identify the quality of your antibody products. It is locally made in Singapore with quality and utilize our proprietary technology to improve the product's quality and quantity. It is more cost-effective, which enables small-scale to kilogram level protein productions based on the same expression system for the best consistency and minimal surprise during the downstream step for further scale-up. Other than that, we also apply unique QR code tracking and automation system to track each production orders with no error rate occur. We are able to scale up and finish by same CHO cell system, which will save time to optimize the condition if you want to move on for large-scale productions. We are able to accept all type of proteins, antibodies, species, and isotypes and fragments. The top challenge of antibody expressions projects are insufficient antibody yield, long time to complete, often takes several months, budgets overruns. To leave out these bottlenecks, GenScript have launched our proprietary TurboCHO platform, which offer higher yield performance within a shorter time period. This is the overview of this platform. They are actually divided into different packages, which are CHO High Throughput, CHO HD, CHO Express, and CHO HP, based on the targeted amount from milligram to kilogram, with a wide range of QC analytical tests available. In this platform, we are able to accept all kinds of proteins and antibody formats. Our PhD expert scientist team will evaluate every request from our clients to ensure the efficiency and quality of the final delivery. What customer need to do is just to provide us the antibody sequence or protein sequence, and we can all the way start from gene synthesis, codon optimization, plasmid construction generation, and transfections of the to TurboCHO and go through the purification and QC check. Ultimately, we can deliver the antibodies and protein in a very short turnaround time. We have successfully expressed and delivered many different antibody formats with high purity and quality in the TurboCHO platform, such as standard typical antibody, bispecific antibody, FAB, single domain antibody, SCFV, and BITE. In the typical antibody drug discovery process from lead discovery to clinical trial and commercialization, we are able to provide different package of TurboCHO platform to our customer based on their needs. We are using the same CHO K1 cell line derived from CHO K1 derived cell line in early R&D and preclinical to clinical trial in our sister company ProBio. Therefore, they will have the same activity and post-translational modification, and our client don't need to worry too much on the expression profile if they would like to scale out and express the antibody for clinical trial usage. We have also performed some in-house expression yield validations testing in some typical antibodies like human IgG1, human IgG4, mouse IgG2A, rabbit IgG, and to track the in different skill package. So the average of the expression yield is relatively high in different skill package. So for this TurboCHO package, which is the large-scale productions, we have repositioned this package to a stable cell pool service, which client can store the pool at our GenScript. We also have done some humans and mouse antibody expression titer tracking between our previous mammalian expression system and proprietary TurboCHO HP system. The average yield for the TurboCHO HP is around 2 to 2.5 gram per liter. The platform enables more rapid and cost-effective evaluation. Besides that, the beauty of this CHO HP service is able to express comparable titer with a stable cell pool with only half of the turnaround time, which is unique in the market. In summary, we are able to provide a rapid antibody discovery platform. Our MonoRab platform enables researchers to generate high-affinity antibodies in a relatively short amount of time, used for therapeutic leads, companions, diagnostic, and reagent tools like antibiotics antibodies. Besides that, we do provide a faster proteins or antibody expression and purification platform. Our TurboCHO platform can be used for fast protein production to generate next-gen biotherapeutic research. This cost-effective production platform enables small-scale to kilogram level proteins productions with a high level of consistency, making it good complement to current target discovery and preclinical pipelines. Therefore, GenScript is able to provide a faster timeline service for discovery to clinical evaluations of novel recombinant antibodies for medical use. Thank you for your attention. If you would like to know more information about any of GenScript service which I mentioned just now, please feel free to contact us directly, and you can scan this QR code to access to our GenScript webpage to get more detailed information as well. Thank you.
All right, thank you so much, Dr. Tan, for your wonderful presentation. So now it's time to answer the questions from the floor. So for all the attendees out there, if you have any questions regarding Dr. Tan's presentations, please feel free to type in your question in the Q&A box, and we'll answer it now. We have a few questions from the attendees, so I'll read them out to you, Dr. Tan. Okay, the first question is, what is the quality of antibody and protein products that's synthesized in Singapore?
We have actually done multiple testing and checking on the proteins and antibody products compared with our China site facility. The qualities of the proteins and the antibodies synthesized in Singapore are the same as what we synthesize at China site facility.
Okay, thank you. So you were mentioning TurboCHO. So can TurboCHO cell line and plasmid be transferred to the clients?
Oh, okay. This question is the proprietary TurboCHO cell line and the plasmid vectors are actually newly invented by GenScript with high yield and high quality. Therefore, we are actually unable to transfer these to clients. If client would like to have the plasmid, we can actually transfer the plasmid to the IP3 vectors and send to our clients. We will also keep this TurboCHO construct with us for 3 to 5 years, so our client can come back and resynthesize them for the same antibodies and protein as well. Thank you.
Okay. The next question is, what are the positive screening assays that used in Beacon for antibody selections?
Good question. So Beacon screening assay for antibody selection is actually include like antigen specific bit space assay, multiplex IgG capture assay, cross reactivity, GFP reporter cell assay, blocking and functional assay. Actually, it utilizes those 4 color panel, which are the DAPI, FIXI, and the TX rate, and the Sci-Fi 404 as a secondary antibody to minimize the overlapping. Based on the brightness level of the 404 to validate the expected targeted expression level. Thank you.
Thank you, Dr. Tan. The next question is, how do we enrich antigen positive B cell before the screening process?
What we have done in GenScript is we actually use a special disk coated with a specific antigens to isolate the antigens positive B cell. The purity of the enrichment B cell is around 40 to 70%. We call these methods antigen panning. Yeah, thank you.
Okay. The next question is, how to improve the half-life of bispecific antibody without Fc while retaining their original bioactivity?
Okay, good questions. So we actually can increase the molecular weight by multimerizations of the antibody fragments. like with the peptide linker or attachments of others molecule, like human serum albumin, PEG, and HPMA. There is also a studies on the carcinoembryonic antigens with CD3 attached with the PEG or albumin binding domain. Even increase the size, but it also doesn't affect the penetration rate. Also, thank you.
Okay. So another questions from the floor. I know some system can hit 5 to 10 gram per liter. So could your system achieve this amount?
Yes, we in our TurboCHO system, we are able to start from very low, which is like 0.1 milligram to all the way to kilogram level.
Okay, that's great. Okay, maybe we give the attendee sometimes to type in the questions because I'm out of the questions from the floor now. So let's wait for a couple of minutes. So
Thank
for
you.
all the attendees out there, feel free to answer. Sorry, feel free to type in any questions. Yeah, this is the time. All right, I guess there's no more questions from the attendees. So thank you so much, Dr. Tan, for your wonderful presentations, and for all the attendees. Thank you so much for participating in this webinar. Feel free to scan the QR code on the screen to understand more about GenScript. Thank you so much. We hope you have a
Thank
nice day.
you.